Hello.
May I request our IQAC coordinator, Dr. M. Sunita Madam, come on to the dais. Now I request Student Union Coordinator, Dr. K. Neeraja Madam, to kindly occupy her seat. I request our in charge of the department, Ms. C.H. Priyanka Madam, kindly come on to the dais. Now I request dignitaries on the dais to lighting the lamp and garland the photo of Srimati Kandikuri Rajalakshmi Garu.
Hello. Hello.
Prayer song by 2nd BSc Zoology students. Now I request our Vice Principal Madam to deliver the remarks of today's webinar.
Distinguished speaker and esteemed participants, a very good morning and warm welcome to you all. It is my pleasure to begin this international webinar on data-driven insights from quantitative genetics and bioinformatics to better decisions. I take this opportunity to thank our Honorable Commissioner, Dr. Narayan Bharat Gupta sir, Joint Director, Dr. C.H. Krishna sir, and Regional Joint Director, Zone 1 and 2, Dr. P.V. Krishnaji sir, for permitting to conduct this international webinar. I appreciate the Department of Zoology for taking initiation to organize this webinar. Congratulations to the organizing committee. Regarding this webinar, data-driven methods are rapidly revolutionizing modern biological sciences, enabling faster, more precise and evidence-based decision-making. The webinar Data-Driven Insights from Quantitative Genetics and Bioinformatics to Better Decisions highlights the importance of integrating quantitative genetics, bioinformatics, and modern breeding approaches to effectively interpret biological data for improving human welfare. Quantitative genetics provides the foundation for understanding complex traits influenced by multiple genes and environmental factors. Bioinformatics strengthens these insights by offering computational tools to analyze large-scale genomic and multi-omics datasets. Hence, this webinar aimed to inspire, create an awareness among data-driven insights from quantitative genetics and bioinformatics to better decisions. Before concluding my words, I would like to send me my appreciation to the keynote speaker, Dr. Mahendra Anumalla, Scientist 1, Late Maturity Breeding Program, IRR Institute, Philippines, and resource person, Dr. Nutalapati Srinivas, Lecturer in Zoology, Department of Zoology, Government Degree College, Ramachandrapuram, Konaseema District. I hope this webinar will be useful for the participants to inspire and create an enlightenment on this topic. Wish you all the best. I once again congratulate the Department of Zoology for organizing this international webinar. Thank you.
Thank you, Madam. Now I request Ranvita, first PSC Zoology, to deliver the introduction of Mahendra Anumalla sir.
Good morning, everyone. It is my great pleasure to introduce Dr. Mahendra Almulla, a respected professional known for his dedication and knowledge. Mahendra Almulla was born on 1986 in Dullur village, Jetiara district, Telangana state. He has completed primary and secondary education from government schools and colleges. He completed MSc in Biotechnology from Usmania University, Hyderabad, Telangana, and PhD from Utkal University, Bhubaneswar, Odisha. In 2009, he joined as Senior Research Fellow at Biotechnology Laboratory Crop Improvement Division, ICAR, National Rice Research Institute, Odisha. His research expertise is extensive and focusing on various aspects of crop improvement such as rice salinity and drought tolerance breeding, genomic regions and underlying accelerated genetic grain in rice alliances and green super rice, impact of the weather and biomedical parameters on sheath blight diseases. He developed methodologies for the data analysis and visualization in crop science. He has published over 44 research and review articles in esteemed international journals and authored 8 books. He is an additional board member of International Journal of Agricultural Science and has reviewed several articles. He attended various national and international workshops. He attended 25 novels protocols available at IRIS website. He attended various training programs conducted by international organizations. He guided 5 MSc students and 1 PhD student. He provided training to nurse, scientists, and students. Dr. Mahendra Anumala is admired for his great commitment towards excellence and his inspiring approach to work and learning. We are honored to have you in the session. Thank you, sir.
Thank you, Ranvita.
Good morning to everyone. Can you hear me? Just anyone just confirm to me, are you everything is audible from my side?
Yes, sir, you are audible, sir.
Great. Thank you so much for the nice introductions from Ranvita. And first of all, good morning to everyone. And thanks for this joining this webinar. Actually, they gave me the brief introductions and highly appreciate for that. So actually here, I'm basically from the breeder point of view as a plant breeders, working on the mainly long durations rice segment, particularly for the India, South Asian, and African countries. So before going to that, our topic, so I would like to first of all great extensive thanks to the organizing committee members, convener, secretary, for inviting me this webinar. Especially, I'm thankful to Dr. Shivaranjan and head of the department, Dr. Raghav Kumar ma'am, giving this invitations. It's a great pleasure and also honor to be present here with you all, sharing to this kind of webinar, particular for this data-driven insights. So as you many of aware of that, because of many of them are, I was thinking to the last couple of days before with Shivaranjani also. So what should I give the best way of a topic so that can be relevant and most of them can understand and engage, rather than giving my more expertise area. But I was thinking in a broader way from the basic to the advanced level, where I can have that expertise skills from the traditional, like an oldest breeding concepts to the modern breeding concept, how it can be useful in the breeding communities. So this topic definitely will give you some sort of insights into you all, specifically for the quantitative genetics and bioinformatics. How will be integrated to take the better decisions to it is because of you all of in the UG and PG, it's not only plant aspect, it also aspect in the animal sides. So how we can integrate to better way of to take the decision that we can cover to the my topic here today. So just to give me this changing my slides. So here I've divided my topic in 5 aspects. So these aspects will you each one of the aspect or sections will be providing some insights wherever you are not understanding or familiar or some of the things are new words or terminology. Try to ask me or try to stop me so that we can have in the same page, we can understand to each other. So this
వెబినార్ ఒకటి
so my sections are first one.
బయోఇన్ఫర్మాటిక్స్ పెట్టాను.
Is there anyone is make it a mute, please? So my section one is section two will be the why we need to do this data-driven insights. What is the major challenges and what are the possible solutions we need to do and how we can integrate with this quantitative genetics. What is that quantitative genetics? So most of the things are quantitative genetics is not aware. We are all of us know specifically the subject for genetics, like in a botany, zoology. We are all knows that, but genetics nothing but to know that genetic information which is coming from the other genes or QTL, that thing is inherited from our parents to the kids. So that is the basic of science, that how it can be understand from the parents to the children or plants to the inbreeds, how passing to the next generation to another generations. So third section will be covering to the bioinformatics. So that's where we'll integrate this, the where we are taking the genomic informations or DNA or the phenotypic information, how we can combine together, make a good decisions. So some of the insight, fourth section, I will give the realistic data tools and methodologies, the approaches currently we are using in the modern breeding programs so that the data can be useful to take the better decisions. So that will be cover my topic in the 5 sections. So as I mentioned, I think I'm not going to this information will describe my the profile, but I want to highlight the things. The area of currently is working in the is the latest technology, like in a modern breeding approach. Because of I came from the plant breeding as a basic a traditional breeder to the modern technology, how we can use it in the breeding program, like in a genomic selections, how to use the genetic gain as a prediction breeding program. Like if you have some data, if you do that breeding program or creating a new variety, that variety will come to the farmers or using to the breeding program, maybe take for the 10 years. But can we predict? Means if you're having the good data, using the data information, can we be able to predict for the after 10 years, what is the impact of that? In that angle, we are working on the predictive based breeding programs. So so with that, I'm going to start our first sections. This is why do we need crop improvement? Because of the crop improvement is nothing but the grain yield. The grain yield, if you improve that, the farmers will get benefit either in the income, either in the food. Because of these are the two things most important in the current world. If you see this crop improvement from the challenges to solutions, I'm highlighting to the challenges. What are the challenges? We need enough sufficient food. Do we have enough food? Because of, if you see this, the population is day by day is increasing. Now early China, now we're in India, the population is higher, but whether do we have enough food? Because the food is going to be down. Why? Because the climate change. The climate change, it comes under many factors, either abiotic or biotic. So when it comes to the abiotic, we call it as a salinity, drought, cold, and temperature. These are the factors will be declining the grain yield. So what we need, current situations, uh, can you somebody else, can you mute yourself? So what we need, so if you see that in the graphical representations from the 2000 to the 2025, if you see that the grow rice seed productivity, now we are having this is 2050. Do we have enough productivity? Absolutely, we do not have enough productivity because of in India we are in the first place, but across the global level, we are in not that level. So approximately we need more than 40 to 50% of the rice production is required compared to all other crops. If you see here, the below of that green color, pink and blue color, the blue color indicating from the rice productivity from the 1970 to 2050. You can see that there is a biggest gap. Because of this gap, it's coming from the the yield decline. We are not that good amount of the grain yield because of the climate variability facing a significant challenges. So with that, our target is we need to produce 1.5 or 2% above the genetic gain. The gain means improvement. Either the improvement is coming from your breeding program. So the breeding program, if you're creating a new variety, it is coming from the 5 years, 10 years, or 20 years. The genetic gain was calculated from the year from 10 years to 20 years or 5 years of the breeding program. That breeding program has to be improved more than 1.5%. But in the current situations, in the rice breeding program, particularly for the different ecosystems, if you're taking from the 20 to 25 years of the breeding program, we are in the position of only 0.1 to 0.2%. Now the challenge will be here. We are in 2025 and we are talking about 2050. Can we able to reach the 1.5%? Absolutely not if we are going to the oldest traditional breeding methodology. If we need to bring that latest technology, particularly the data-driven insight, so that we can improve the grain yield. So that's where I'm explaining that in the 1950s, 1960s, all of us know that green revolutions. That is coming from the traditional breeding program because of at that time we do not have any technology or any data. The breeder has to choose the plants in the field by visual observations. So later on, it's coming from the egotype breeding program. These are different population improvement breeding program. Now we are talking about the predictive breeding program. So this is the journey of the traditional to predictive breeding program. These are the only situations or solution, the breeding strategy only we have. With this, the only expectations, hopes for the data. If we have the very good data and very good expertise, the data analytical skills, then how we can integrate this different data points to make a better decisions. So that's our main topic of the presentations will be here. The data is the only fundamental source from coming from the your any data, either coming from the environment, either coming from the phenotypic or genotypic informations, how we are integrating together to take a better decisions to develop high yield productivity and also developing new varieties that can be resistant to the either drought, salinity, flooding, any abiotic and abiotic stress conditions. So coming to this, whether why these data driven are more important, because we are talking from these challenges, solutions, and opportunities and data. What are the data types are available in the current scenario? So now look at from the your any scientific community or any research community. There are a lot of so much of data has been developed from the biological informations. Now we are studying from the biology. So biology is not only one aspect of the plant, either coming from the botany, geology, chemistry, and physics. All of us comes together, then we can identify that solutions, how these all subject integrated together to make a better decisions. Any subject, we will get some kind of data. Either the data is coming from the either your numerical value, either characters, or some kind of logistical informations. So these data informations will be generating from the lot from the our olden days, like in the 1970, 1960, or 2000. The technology has been developing from the rapidly and modern way. The data house is emerging a lot. But do we have enough sufficient data, or do we have enough expertise skills on that, that is still it is in a gap? So the data is generating from the lot informations, and how we need to take that decision, it is still it is the biggest challenge in the any breeding community, is a plant or breeding. So that's where the data driven conditions like in a machine learning, artificial intelligence, that comes into the place. So now the question here, Whether do we have adapting the standard robust statistical approaches in the research? Absolutely, if you are integrated the standard robust analytical tools in the breeding program, then only we can have achieve the sufficient whatever the goals. So that now the question is the sound data driven framework is one of the most important to understand what type of data available with us, why effectively data quality and appropriate statistical methodologies and strong biological insights are mostly needed to identify from the your better decisions. So as I mentioned, from observation to predictions, because of earlier plant breeders, what they will do, they used to go to the field. In the field there are a lot of plants are there, so which plant looks good either in the biomass or in the grain yield. So based on that, they will go for the some visual selections. But now we are moving from the data-driven modus, which means we are collecting the data, using the data informations, how that particularly rice variety or any crop going to perform after 5 to 6 years in the field. We are going to predict. So that's where the data driven or data science playing a major role in the any scientific community. That's where our topic is what kind of data available with us, how to integrate, what are the approaches are available in the scientific community that we are connecting together. So now moving to forward, what kind of data types are available with us? Now look at this. I'm talking, as I mentioned, as a plant breeder. So most of the things are my data. It's related to the crops, but these all things are well correlated with the plants or animals because of I'm. These slides are very basically understand from your all as prospective way how integrated these. What is the quantitative genetics? How this bioinformatics will help us to take the better decisions. So now coming to these data types, data is not just a number, it is the integration of the multiple data type points to make the better decisions in any scientific community. Now look at see in the center of the database, this is the informations comes together all from coming from your phenotypic, either genotypic informations or environmental informations or the special variants coming from the field. This comes together and database will take the decisions. So let's talk for the phenotypic data. So what is the phenotypic data? So in the traditionally we measure the plant height in the field at the flowering time, because when the flower it's formed, then it's when it starts from the seed setting, and what is the plant height during the reproductive stage. These are all this some kind of phenotypic informations which are measurable traits from the field. Second is genotypic data, which is coming from the right side. The markers are single nucleotide polymorphism. These are information generated from the different sequencing technology from any crops or any kind of animals. So third point is the particularly environmental factors. The environmental factors which includes either soil parameters, for example, what is the soil fertilizers, soil organic carbon, what is the nutrients available in the soil, and temperatures, what is the cold temperature and the high temperatures, and what are the flooding events. So these are all the environmental factors. So fourth will be the special variants which is coming from the word field. So when the plants are grown in the field conditions, we have the planting in the field as a followed by systematic way as a field row or column informations are the replications. So this information captured. Hello. Can you hear us? Is it okay? Is it audible from my side? Okay, thank you. So now the question is how we need to integrate this kind of data informations into the database. From there, we need to take the decisions whether the plant has good performance or bad performances. Because of earlier in the traditional way, just we are taking the traditional way because of we do not have any technology or data analytical tools. That's where for generating any single variety, it's taking a lot of years to bring the new variety to the farmers. So the data playing a key role. How do we analyzing the data? How do we take the decisions? So now the key message of this slide is in a modern crop science, the connecting these data types to make the breeding is more predictive, efficient, and impactful. And when we capturing these data types in a phenotypically, genotypically, environment, and field, are we facing any challenges? In this case, yes. We are going to face a lot of challenges when you're taking these informations to make it as a data-driven decisions. So it's, as I mentioned, these are all some of the key challenges to take that breeding or any data-driven decisions. So first I'm highlighting these complex traits. What does it mean, complex traits? So complex trait is, which are one of the most biological traits. It's coming from the polygenic traits. If you remember from the Mendel principles, there are some kind of monogenic and polygenic. Polygenic means there are a number of genes or QTLs contributing to the expression of a specific trait. So that is interpretation is very, very difficult because if it is coming from the single genes, we can decide that whether it's a phenotypic expression is either color, either plus or minus. But when it's taking from the grain yield, and you're improving that grain yield and which is coming from the several genes, it's not the single genes. So how do we capture that variability, and how do we take the data driven decisions for the complex state is one of the biggest challenge. Second is environment variability. So now there are many varieties or many plants are developing and improving. When they are growing in the field, there is a huge interactions with the G by E. What does it mean, G by E? Genotype by environment. Because these genotypes are performing based on the environmental conditions. For example, in specific for Andhra Pradesh, in Rajamandri, if you're growing a particular variety, that same variety growing in different state in Karnataka, whether we are getting the same yield or same performance, absolutely, it's not, because of there might be the soil profiling, the humidity, the temperature will be different. In that scenario, that plant is having the huge variations. So that's where environmental variability will be creating a huge disturbances in across the seasons as the locations. Third is huge data sets. As I mentioned, the data is generated from the in the different ways. The phenotypic is coming from the field, or the phenotypic coming from your greenhouse. The phenotypic data is coming from your high throughput phenotyping using the some kind of sensors. So whereas in genotypic information coming from your Sanger sequence method or yours GBS sequence or 1K what there are different types of sequencing methodology, and this methodology is providing the lot of data points in the genotypic informations. So third will be the we need for the speeds means the traditional breeding or selections is very, very slow to generating any new variety. I will come to that particularly the need for the speed, what are the traditional breeding concepts. So in that case, we need to have the modern breeding program, and we need to have this systematic way of data points. And fifth is the foremost important is the lack of skills and integrations. So how to integrate these different data types coming from your environmental informations, genotypic informations, phenotypic informations come together. How do we analyze in a robust way to take the better decisions? So this is one of the biggest challenges in a data-driven breeding program or data-driven insights. How do we overcome of these challenges into the modern breeding programs? So that will helps to understand that. So what tools can be useful in the community? I will cover into the fourth slide. So with these challenges, I'm going to explain that how it is a traditional breeding methodology. Why do we go for the modern breeding program? Because of, as I mentioned, there are many challenges and solutions and opportunities. We need to produce more enough food within the short period of time. At the same time, we need to utilize less resources. In the resources means in terms of as a lowest money within the short period of time, we need to use that or develop that new varieties. So that can be bring this technology, then only we can bring that higher productivity. That's where we want to have more than 1.5% of the genetic gain to sufficient to feed the global populations, or come from the climate changes. Now, the question is, as I mentioned, why do we use modern breeding tools? Just see this center of that. One is time, one is money. Of course, every one of us running with these two things. So for the UG and PG, for the student perspective, and anyone online or offline for interesting to that, just I made this slide for very basic understanding why do we need a modern breeding program. Just imagine, left side is you are in the original and destination. Let's take example. I am in Hyderabad, just I want to travel to the Rajahmundry. And at the same time, there are many ways. I can come by maybe by boat or flight, by road. But it's all depending on the how within the short period of time and less, because of flight will be there within a 1 hour I will be there, but it is expensive. Road will be there, lesser amount, but it takes time. By ship, it's the same thing. Similar way, just I'm correlating with that breeding program. Here it is the parents. The parents is coming from any kind of your crop. Just take any example, either your maize or cereal, sorghum, chickpea, rice. These materials coming from your elite breeding lines, or maybe the released varieties, or any kind of traditional varieties. The breeding program is one of the main objective. Want it is to develop the products. The product is which is better than the previous one, and that will be giving that a higher yield, so that the farmers will get a higher yield productivity. At the same time, the farmer will get benefit. So we have this conventional breeding methodologies. So this is conventional breeding methodology. If you go for the one parent, one variety to another variety crossing and get the hybrid, and go for the segregation line to fix the homozygosity. When you look at the breeding program, when you do the crossing with one parent to another with the pollen and anthers, it get fertile, and that will be the ratio we all of know, that F1 50:50 ratio. But when you go into the next F2 generations, it will be going to be segregate. So to make it homozygosity, it will take long time, maybe the 3 to 4 years. And once it is fixed, it will go for the on-farm trial testing or multi-environment testing. After that, that will go for the releasing system. Currently, what we have that ECRIP or National variety analysis system. But that will take for the 10 to 12 years. But the question is, we are not that in the position following the same method. We are want to have the modern breeding as we talking about the data science or data driven insights. Are we talking about the speed breeding? And we are talking about the some modern breeding program or modern breeding strategies like genomic selections, population improvement breeding strategies. I'm suggesting to these, take note of the students when I'm saying this data science, population improvement breeding strategy, speed breeding. So if you remember these words, whenever you go to home, just have a search in the, everybody's having the mobile might be. So search that, so how this tool effectively can be used in the breeding program to develop the products. So that's where within the 3 to 5 years of the years, we can develop the new varieties. So this information, how do we connecting with the bioinformatics and quantitative genetics? Now, as I'm going to be go for the foundation of the genetics. Genetics is nothing but is which is coming from the genes or QTLs, and how this genetics was born in terms of in the historically. What are the phases of the breeding strategies from the any crops from the 1960 to 2025? What are the phases, and how do we look for the data in this way, and how to make it as a omics approaches to make that data analysis will be covered in the fourth sections. So this giving you some basic understanding, this basic quantitative genetics is nothing but, as I mentioned, is a branch of genetics. So any traits in the plants, either plant height, grain yield, these are the traits which are coming from the associating with the specific some genes, either maybe single genes or many genes. So here there are two things. We need to understand qualitative and quantitative traits. So the qualitative traits is nothing but simply encoded by the monogenic traits. It's single allelic variations that giving the color variations. So look for the qualitative, there are flower colors, either orange, pink, or white or purple, but that is encoded by the single genes having the allelic variations. Quantitative traits, like example for the plant height. The plant height will be the different, but that is not encoded by the single gene. It's a continuous variation. So if you see in the graph, this is the one plant, each graph is one plant, means the plant height will be the different, but the distribution pattern is encoded by the genes or QTLs that affecting by the your quantitative traits. So the qualitative traits easily we can see in the from the field by visually, but quantitative traits we cannot see. As a biologically, there is a inside the genetics will be contributing for the your specific traits. That's where the formula is coming here. P equal to G plus E plus G by E. P is equal to your nothing but your phenotypic. G is your genotypic informations plus G into E means genotypes by environment interactions. This will be providing that how the plant shape, how the plant architecture would be in the field. On the left side, DNA or genes are QTL represented from your genetic contributions. Whereas in the right side, environmental factors like in the soil, temperature, water, climate, these are all environmental contributions. So now quantitative genetics as a bridge between the genetics, phenotypic and field environmental informations. This will be very, very essential to understand the modern breeding and bioinformatic aspects. So without understanding these genetic variations, any data types, any data analysis, that will be leading your misleading of your output. So therefore, without understanding of this quantitative genetics, proper statistical analysis, and knowing that how this genetic and environment influence of the traits, we cannot go into the take any better decisions. So the take home messages from here as a bridge between bioinformatics and quantitative genetics. This is most important, and just for you all students, I think this is the one I cannot identify any literature, because of this is a modern quantitative genetics. If you see that Bartha quantitative genetics from the 1860 to 2020, how this genetics evolved from the current scenario. When you look at from the Mendelian genetics, it has started from the 1860s. There he was explained that the how these traits are inherited. At another one is evolutionary genetics that led by Darwin, who's explained that the species changes over the time. So another track below that, that is Pearson, Weldon, and Galton. What they mention is these are all the continuous traits which are coming from the plant height, yield, or weight. These are called as a biometrical genetics. So now we are established in the 1860-88 biometrical genetics, Mendelian genetics. These branches come together, and they found that in the 1900, Fisher and Wright and Holden, they demonstrated that these many genes, which called as a polygenes with a small effect, could produce the continuous variations. So Mendelian said that the single gene will be affecting the single trait, like in a way, the flower colors. But these biometrical genetics, they peoples, they said many genes, any trait will be governed by the many genes that can be contributed the trait of expressions. But this groundbreaking foundation of the quantitative genetics in 1900s, Ronald Fisher, Sewell Wright, and Heldon, these are the stalwarts of the quantitative genetics. That's where it started, from the combining your molecular genetics and quantitative genetics. That's where lead it to the what is the relationship between the your trait and genetics. That's where we call as the quantitative trait loci. Now we are talking about the quantitative QTL mapping. So later on, when that 1950 DNA was discovered by the Watson Crick. So again, there are DNA is a genetic material, non-genetic material. There are many hypotheses. After getting that informations, they identify the some kind of technology has been developed, some statistical analytical tool has been developed. This started from the genome wide association studies, or GWAS, that has been built into the identifying the many genes which are associated with you with your trait of interest. So that's where the foundational quantitative genetics has been discovered in the 1990s. Later on, it's moved to the QTL mapping, Jiva selections. Now we are in the state of genomic selections. That was first they published in 2010. So the genomic selections is one of the breakthrough um, breeding strategy, where we can identify the best of the best lines we can use in the breeding programs. So that's where, from the evolutionary Mendelian genetics to the modern synthetic biologies. So that's where journey of the quantitative genetics. So this is the same way explain that the earlier the ideas coming from the Mendelians and evolution from the Darwin, foundation of the population genetics, which is explained that how these genetic variations is maintained from the your parents to the offsprings. So these concepts expanded into the quantitative genetics, molecular genetics, genomics. Together, they formed as a modern synthesis that drives today like QTL mapping, GWAS, or genomic selections. So this is just for your understanding, for particular for the user students. The modern quantitative genetics, the people behind of this, what we are talking about right now, the latest technology, the latest information. These are the founders, or these are the legendaries of the quantitative genetics. So first one is Gregor John Mendel. He talking about the how this inheritance means from the parents to the kids, because of everything is coming from the your genetics. So how this genetics passing to the single gene inheritance. Fisher has found that this is not coming from the single genes. There are multiple genes coming from your parents to your kids, from your kids to the next generations. Because of that's where we are talking about the additive dominance genetics. Because when you having the early generation, there will be dominance will be there, but later on, when you fixing the homozygosity, there is only genetics that is called as the additive genetics that is only passing to the next generations. That's where in the modern breeding biology, how do we capture this additive genetics? Take the note what is the additive genetics, how it can be useful in the scientific community. Just search in the Google, maybe you can have some information. So later on, the pedigree relationship matrix is nothing but it is taken from our parental informations because of the parents' information uh, as A and B, that information's keeping into your data analytical models so that we can capture that what is your next generations of your plants. So like that, the evolutionary, I'm talking about these genomic predictions. Currently, we are working on these aspects. This is not a new, but that's a foundation led by Matthew Vision from the 2001, particular from the animal. So now we are implementing into the particularly plant breeding communities. So now as I mentioned to you, the breeding community or breeding strategies, it's not the new. I'm trying to cover for your UG aspect level. So there is from the phases from the traditional to modern breeding programs. If you see from the just I'm highlighting from the 1900 to 2000 in the below. So 1900 to 1980, the period we all of us know, the MS Swaminathan, Norman Borlaug, that's a green revolution time, where we double that granular productivity because of the only thing is we followed the pedigree, parents do the crossing, and we're selecting the lines based on the visuals selections. So later on, some kind of SSR markers, RFLP, AFLP, different STS markers has been developed and use the trait-based breeding program. So based on that, identify some plants by genotypically. Because of that, markers or information is confirmed by the recombination frequencies from the plants. So that will be using the genetic informations, identify the specific lines to take the forward to the next generations. So modernized breeding program, as I mentioned, 2001 onwards, the genomic selections foundations has been made because of past breeding program. We are talking only for the specific line, specific one or two varieties will be selecting from the first generation to the next generations. But in the modern breeding, we are not talking for the only one. Not talking from the only one variety. We are talking about the total populations. Because when you do the crossing A1 and B, one plant to the another plant, we are getting the hundreds of lines. What is the variations coming from the your parent one and parent two? That variations will bring into the genomic selections. So that is we called as a covariance between the varieties. So that's where the past breeding program 10 to 12 years and we are modern breeding program 3 to 5 years to bring the new varieties. So this is for the just giving the some glimpses. This is a more advanced, but just for the faculty or anyone, maybe online students also may be interested. What are the mapping? Because of any student, they are working on the QTL mapping or genome-wide association studies. So what is the ways to map the genes or QTLs? So means, as I mentioned, the quantitative genetics nothing but capture the genetics, either the gene or QTL that is associated with your phenotypic trait. The phenotypic trait, anything, either disease resistance, plant height, grain yield. But the phenotypic expression is contributed from the genetics. The genetics nothing but biological information which is coming from the inheritance pattern. So the DNA. So from there, how it is passing to the one generation to another generation, that is only from the recombinations. Let's take example here. So here, the first one is there is a one parent blue color, another parent is red color. Now we did for the crossing. And when you see the generation, next generations, the recombinations is happening from the your chromosomes. Let's take an example. Rice is having 12 chromosomes. So in the chromosomes, one parent to coming taking the higher, or other parent chromosome will be the lower. But something the recombinations will be happening here. Based on the regressions, again the data analysis comes to here. So now I know this parent, what is the phenotypic informations, what is this babies, like in a parent one and parent two. Now we are, when you go for the generation, we have so many babies, all the babies coming from the same family, but all of them are different genetic constitution of alleles, genetic constitution of genetic variations. Because the genetic variations we cannot see visually, the variations we can see either from your color, either from the plant height. So something, the variations will be there, but that genetic variations we cannot calculate it until unless if you have the genotypic informations. So this advantage of this mapping methodology, we can identify that particular your grain yield trait associated with these markers. We can go for the QTL mapping. Certainly, if there is no any, because of these two parents are already released varieties or fixed populations, whereas diverse populations means some kind of traditional variety, maybe Rajamandri, Chittamutyalu, some other traditional varieties which are which are having very good traits, but the yield was very low. So how do we capture that genetic variations into the breeding program? So this is called as the GWAS, genome-wide association studies, where we can identify the ancestral recombinations events, because any recombinations will be happening from the any diversified plants. But this one, the recombinations will be happening only within your two parents. But here, let's take example, if you're having the 50 parents from your traditional varieties, this has been generated from specific regions or specific geography, but all of them are genetically having the variations. That variations will capture that using the diverse set of populations. So third one is a nested population. We call it as is using the different donors or different elite lines. So capturing for specific QTLs or specific alleles responsible for the your trait of interest. So this is kind of a methodology will be implemented to the traditional to the modern breeding. All of the things will work on the only one and only aspect is regressions. The regressions data analysis on the phenotypic informations and the genotypic informations. So the genotypic informations coming from your sequencing, and phenotypic information whether coming from your field data, either you're coming from the high throughput phenotyping. So just I'm highlighting one of these figure. Let's assume for the chromosomes, rice is having the 12 chromosomes. So now one of the chromosome will be here, and the bars, different bars indicated from the your some segment of the chromosome, like in your genes or QTLs will be specific markers are located on the chromosomes. Now I'm going to this particular for the traditional breeding methodology. I mentioned the conventional. Later on, it came to the marker-assisted selections or marker-assisted backcross, marker-assisted recurrence selections. So this methodology will be tell you that these markers, with these methods, these markers are associating with my grain yield. But wherever the trait associated, only those markers will be giving that information. But rest of the other colors, like in a black, red, yellow, it's not capturing the marker assisted selections. This is your trait of interest. That is the marker assisted selections or breeding program where we can do the QTL mapping. Second option is GWAS. This one, genome-wide association studies. So what it is, this method simply is, we can get that whole markers informations. When you're regressing on your phenotypic information and the genotypic informations, we'll give you that which genetic marker significant, which are non-significant will not giving that information. Means The light yellow color, 1, 2, 3, 4, that will be giving to the jivas. Marker resistor selection also giving the specific. But now still we are missing the some genetics here and some genetics here. But genomic selections will capture the all kinds of markers integrated into the your breeding program to take the better decisions. If you remember, the breeding strategies, all recombination events coming from the additive dominance and epistasis will be there, ADE. But when you're breeding from the long-term breeding program, the only one thing is additive genetics. That is only forward to the next generations. So genomic selection is the most powerful when you want to capture these genetic variations from entire chromosomes. This only for, I'm mentioning for you all example, 1 chromosomes. Similarly, all remaining 11 chromosomes, we are capturing the genetic information of this, all the markers. That's where we are getting more powerful than marker resistant selections or JVAs. That's where the breeding value will be more robust. So now this is again similar to same concepts, but thing is, how do we choose that genomic methodology based on that marker density or population type, or what are your main trait? So let's take an example here, the low density marker with biparental populations that leads to the QTL mapping. Biparental multi-parental population leads to the QTL mapping. The objective will be identify some genes and QTLs. The methodology, the approaches will be here, marker resistor selections. Second will be the diversity panel, which are coming from your traditional variety. That methodology will be JIVAS. Again, that will be the objective will be the genes and QTL. QTL nothing but is quantitative trait loci, which is governed by multiple genes. And whether this trait will be affected by the small or large, but the methodology of the breeding strategy, marker resisted recurrent selections. The third emerging is using the next generation sequencing technology. That's a very high powerful, using the genomic models, creating the breeding populations, that is called as the genomic selections. So the ultimate aim of this slide, I want to show you that if you remember the first slide, if you need that more food, we need the 1.5% of the genetic gain. So the genetic gain, nothing but if you follow the traditional methods, we will not going to achieve. We have to follow that modern genomic selections, very systematic data analysis, then only we can achieve that your genetic gain. So that's where data capturing information is one of the critical into the any breeding program. So this is also, I'm giving you some insights. What is that marker resisted selections and genomic selection? Just giving you some, I think some of the PhD students or maybe PG students will helps to you. So what it is generally, there are, let's take an example, if you have the 100 genotypes, these are some of the specific SNP markers informations. So the data analysis based on the regression of your phenotypic and genotypic informations will get under some specific significant SNP markers, either SNP 6 and 14. But here totals 15 SNPs are there, but we are giving that the peak is coming from the 6 and 14. Rest of the markers, still those markers are giving the phenotypic contributions. If you remember these marker resisted selections, the phenotypic variants explained by 20 to 30%. But what about the remaining 60 to 70%? That we are not capturing. So that's where one of the limitations from the marker resisted selections, we are missing the heritability. Few SNPs do not explain that all variations. So that's where, how do we capture that? The genomic selections, if you take that, again, same 15 SNP markers, each markers are having this certain contribution to their phenotypic informations. Now we are building a model or statistical analysis based on your phenotypic informations or genotypic informations, and that matrix will be giving that certain values. That is we call as a breeding value or genomic estimated breeding value. take note of the students, what is the breeding value, what is the GEBVs. So this will be you giving some insight. So based on that, our objective will be the which variety is performing better out of this hundreds of lines, which are the top 10 lines will be giving that best yield, so that I can use these parent lines for the in your breeding program. That's why just highlighting the some of the variations or difference between the genomic selections to marker register selections. So so again, this is some kind of holistic breeding program. This is one of the modern breeding solution to improve the grain yield for the very fast manner and a very efficient way. How do we use that? Just keep on eyes here. So this is a funnel diagram, and the first one is I'm giving the name of this base populations, means whatever your plant materials or whatever your genetic materials, those materials are well collected from your ecogeographic region. Let's take example Andhra Pradesh. We all of us know the Samba Masuri, Swarna, some kind of varieties has been very well utilized in the farmer field. So if you want to improve that better than that variety, I need to have the better varieties, maybe 10, 15, 20 or 30 to 40 lines. From that, I'm going to do some crossing. So one variety to another variety, I'm doing the crossing block. So elite means these are the fixed or elite lines, release varieties. So once I do the crossing, the baby plants will come F2, F3, F4 means each generations will go for the here speed breeding or recurrent genomic selection approach. In this place, what we can do, we are fixing the lines as much as faster, because of the here the if you remember the one of the modern breeding aspect is speed breeding. What is the speed breeding? Just take note, maybe you can search on later. So that from there we are getting some certain number of lines from this family, because of here I'm just giving example. One family, one parent is a male, another parent will be the female. From these lines, just getting that next generation to another generation, bringing the around 1000 to 2000 lines. Now the question is, these 2000 lines, how can I identify the one or two best varieties? If you go for the traditional breeding program, it will take for 10 years. As a modern breeding program, because of from here to here 1 year. If you go for the genomic selections, we can reduce the remaining 2 years of the period. So that's where less time, how can we identify the best lines from the 2500 lines? That's where coming to the second one is genomic selections. Means genomic selection, genome means utilizing the complete genomic information to select the best individual lines. So from there, 2500 lines based on the genetic informations are based on the genomic relationship matrix. Means, for example, let's in your class, UG students, you are having the 100 members. Out of 100 members, maybe college wants to go for sending to you some camping, but they want to send only for the 10 members. How do we select the 10 members? Either maybe somebody is very talented in the speaking, or somebody is very talented in the reading, or somebody very talented in the subject. So based on that certain informations, out of 100, 30 members will be selecting for the camp. Similarly, 2500 based on the genetic informations. Plants, all of them are look same, but genetically will be the dissimilar. So that's where the genomic selection, we're creating that 200 or 300 training set. That lines can be evaluated in different states, maybe Andhra Pradesh, Tamil Nadu, Karnataka, Maharashtra, UP, and do that analysis of the phenotypic informations and get the best of the best breeding values. Because when I'm saying the breeding values, these are not coming from the phenotypic information. If you remember the data type slides, the phenotypics, genotypic information, environmental information coming together, that will be giving you that breeding value. That will be most powerful that lines will be used in the again crossing block. Means again to the whatever the best line is coming here, we'll do the cross here. So in this way, earlier, if you are getting the 4 tons per hectare, and the next generation of your breeding cycle, you will expect that 4.5 tons per hectare. Like that, we can increase in the yield productivity from the one generation to another generations. So this is a um, modern way of breeding strategies that currently also we are doing this similar approach. Just take note from the students, what is the genomic selections, what is the breeding values, what is the speed breedings, and what is the recurrent selections. Just take note of these terms, might be useful for you. So the bigger picture of the genetic gain, as I mentioned, so always you used to do every cycle of the breeding program. We need to improve your mean of populations every cycle. So that will be giving that your recurrent selection breeding program. So that means crossing to cross, and recombinations will be happening, and select the best lines from the your field, and again select the past best performance lines and do the crossings. So I think I'm not going to go very brief, because I want to highlight that in the modern breeding program, the how heart is important to the our life. Similar way, the modern breeding program, the heart of the breeding program is breeder equations. Just take note what is the breeder equations. Because of the breeder equations will tell you that how efficiently breeding program you're doing, the selection intents, how you're selecting, how you're identifying the best lines, what is your breeding cycle, and what is the variance was present in their breeding program. So this is the hardcore of the breeding program for the breeder equations. So this means simple is, if you correlate with if you want to fix the door in a window, you have in your hand nails and will be there, but where you want to fix the nails to make it very perfect, you know that where is the gap and where is the height and weight. Similarly, as a breeder, really need to understand these 4 concepts to make it more efficient and productive way. So now I'm coming to touch on the quantitative genetics and modern breeding. Now I'm going to give that some highlights on the bioinformatics. So these are the different types of your plant varieties taken from the phenotypic informations. And this will be from extracting from the DNA. There are many ways to do the genotypic informations, Sanger sequencing, GBS sequence, whole genome sequencing methodology. And the plants, because a plant has expressions is different stages, different growth stages, and different environmental conditions. But what is their expression profile? Either expression is coming from your RNA level. How do we capture that informations? That information go for the transcriptomic sequences or proteomics or metabolomics. These are all your bioinformatics. Bioinformatics, nothing but biological information which is coming from the DNA or your RNA or your proteins. This information will make it stored in the database. That's where bioinformation will come, taking the phenotypic informations, bioinformatic informations, quantitative genetic information to take that better decisions to in a data-driven insights. So with this, some of the statistical tools I'm giving that informations. So those will be some kind of variance and also some of the correlations and regressions. This will be the fundamentals of the statistical analysis. As I mentioned to you all, just take note what is the single trial analysis and what is the multi-environment trial analysis. So this will be based on these parameters only we can identify the best lines, which is giving the best yield. So just want to touch with you one of the point is heritability. So any plant breeding community, any crops, if you're going to see the performances in the field, based on the heritability only we can assess that the plant performance is good or bad, because the heritability is coming from the your how the experimental design, how your genetic informations was calculated. So based on that, we'll assess that particular one location to another locations. Because of that, heritability value will be coming from the either 0 and 1. So 1 is a super and super duper, 0 is poor. But wherever the 0 0.1, 0 0.2, something is there in the heritability which is coming from the one locations, that will be something is a problem. So we need to understand that when you're having the data coming from your any scientific, what is the heritability? The heritability either coming from your broad sense heritability or narrow sense heritability. I'm not going to the very detail, but only thing is for you understand, broad sense heritability is only coming from the genetic variants, either the genetic variants coming from the additive, dominance, or epistasis. But narrow sense is the most important for the breeding. This is only coming from the your additive genetics. So that's only fundamental from the improving the yield. So now, as I mentioned, the bioinformatic coming from the biological data with the different instrumental or different methodology, that can be so that biological data integrated to the data analytical pipelines. This is the one of the most important in the community, scientific community, how we can do the data analysis. The data analytical pipelines, which we are published in the last year, last 2 years before, very systematic way. I think this is the one of the best robust way of data analysis. And what are the analytical tools? As I mentioned, what is the Jivas, predictions, and multi-omics platforms are available. So as I mentioned to you, I will try in the end of the slide, I'm going to give some of the tools, what are the currently available. So when you look at from the Jivas, there are tools like a Plink and Tassel software, highly used in the breeding community. As the genomic predictions, there are some packages will be used in the predictions, like without any testing, we are going to be
లింక్ వచ్చింది సార్ చేస్తాను రిజిస్టర్ అయ్యారా
yeah some of the how we can omics platform technology will be used in the bioinformatic platforms. So these are the very fundamental how to integrate three pillars together in the to take the better decisions on that. So just I'm giving you some of the historical, the traditional breeding, how it is developed, how the markers has been developed, how the quantitative genetics developed. Now I'm going to give some information how this data analysis has been developed from the previous to last 2010 to 2025. So earlier you used Windows-based statistical tools because of we do not have much information how the data is giving, because the data is keeping in the Excel sheets. Like once Excel sheet is uploaded into the software, that will providing some informations. Because of that, earlier used to do that, but now is there are many advanced cloud-based analytical tools like an R Shiny, R Program, Python. These well advanced tools are available in the breeding community. So as I mentioned, these are the previously visual selections. Now I want to take the students, because of blues and blobs are very fundamental into the current technology, how we can calculate this information in the data tools, how we can take the decisions. So blues is the simply taking the above the means, because we are calculating some information from the error variance. Blobs, it means is a best linear unbiased predictors, and this one is best linear unbiased estimators. So this is a very very fundamental in statistical analysis tools, more robust way of calculating and to take the better decisions. So just take a note on these two things, how do we do that. So might be useful if you are anyone interested in the research, or maybe these words may be familiar to you. So I think this is one my last slide on the data analytical process. So any data is coming from your phenotypic or genotypic information or environment data that can be stored in the metadata. There is a many ways we need to do, like in a pre-processing, how to do the modeling and do that output of the results. So this was already published in our team. It was in the online and GitHub also. So if you're not following this specific way, your whatever the taking the decisions on the your data, that will be take you misleading of your selections, or misleading of your the best materials. So that's where data will be providing you more insights. The question, the key message from my side is data driven crop science is completely either biological or environmental or quantitative. It's a very actionable insights. This all comes from the realistic informations to make it as an improvement. That combining the good data, strong analytical methods, biological understanding will provide you a lot of informations. Now the present situations, no single gene or environmental factor will explain any trait on its own. So the breeders or researchers or any professor need to be precise, measure that data types, integrated into the well database, and followed with the mixed models like as I mentioned, blobs, breeding values, and special analytical tools that will be addressing to the true genetic value, which is coming from the phenotypic and genotypic, so that we can avoid that background noisy or the background errors, to enabling the more accurate predictions, or to more uh, identify the best of the best lines. So the combining with this smart data analysis skills and emerging omics tools, the data-driven approaches empower to the breeders or scientists to make that better and long-term decisions. Therefore, to achieve that our real improvement in the crop breeding communities. Because of all this improvement, it's not only for the sufficient food, but most importantly to overcome of these climatic challenges. But there's a climatic challenges coming from the any situations will come. Either heat, now we can say that it's coming to the very high cold or high temperatures, are frequently facing the cyclones. With these challenges, if you're not integrated with this data or historical data information in the right way or right skills, right way of informations, we cannot achieve that better yield, and we cannot take the better decisions. So without just with this, I'm thankful to my leadership, because of when I got this opportunity, I skillfully accepted the invitations and approve my ERI leadership. Because at the top of this, my leadership, my current team, the program was led by Dr. Wasim Hussain, and these are my team members in International Rice Research Institute, Philippines. So right now I'm in here, but I'm very closely working with this HQ, and these are my mentors. Because of my research career has been started in with Dr. CNN Neeraja and Raindra Babu. That's where Indian Institute of Rice Research, Rajendra Nagar, where I started in the last 15 years, and I work with the different legends of the plant breeding teams. So with that, I'm thankful to you. And if anyone PG students or UG students, I'm referring to these books are very useful for your understand this quantitative genetics and modern data analytics. So I'm very happy to have you. If anyone having any questions, please let me know. So once again, thank you for the committee for giving this opportunity for delivering this webinar on data-driven insights. I hope something you will have some idea, because of with the short period of time and bringing this call concept is very difficult. I know some of the UG PG state students, but my major emphasis on that. If you remember some of the terms like genetic, what is genetic gain, what is genomic selections, what is recurrent selections, what is bioinformatics. If terminology, if you some out of this 100 members, if you any one of you going back to home, and if you're searching of any one of the word, I would be very happy, and that will be the my satisfactions to deliver, giving the my presentation on that. So over to you for the Ranvita and Shivaranjani. If you have any questions, I'm happy to answer that. Thank you.
Thank you, sir. Dear participants, the section is open for the questioner.
Very good morning, sir.
I'm Sneha from first year. Question
Okay.
is what? My question is what future of data science because it is dominating right now.
Sneha, can you repeat one more time because of your voice is disconnected in between. Hello.
What is future of data scientists because AI is dominating right now.
That's a very good question, Sneha. Thanks for being that because that's where like because when I heard this webinar, particularly I was mentioning that's data-driven insights because of now we are going to move forward 70%, 80% of the going to the data-driven breeding decisions. But the critical and most important challenge is how accurately, how confidently data you have in your hand, because that will be tell you that because of that reason only we are going to take the better decisions. There is no any doubt on that data science will be playing a key role in the coming days, or artificial intelligence playing a key role on that, no doubt. But how do we analyze? So what are the data inputs you are giving to the software or to the algorithm? Because all these are all the things will work on the certain genetic algorithm tools. Like one of my mentor, he always used to say that artificial intelligence like in a mother. So the mother used to grow that babies based on that experience, how you're going to the babies, how you're going to the feed them. If you're going to the feed them with very good nutritions and a very good informations, the baby all will grow very good stronger. Same way, the artificial intelligence, if you're going to the what kind of data you're going to give them, are you confident about your data informations? Is it realistic value? Is it enough that data is to predicting or to take the better decisions? So that's where need to be very critically, carefully take the data based on the breeding, any kind of better decisions. But in the coming days, every student, if you learning are either going to be the quantitative genetics or any research fields, data science will be one of the most important. Each one of the person has to be aware of that, how the data science like in a R Shine, and now it is coming from the R program based R Python Shiny, because there are Shiny packages are day by day it's coming. So these packages are built in from the many quantitative genetics or data scientist peoples. So there is lot of scope on the data science in the coming days in all aspects, either animal breeding or plant breeding and any research oriented. Lot of future on that, yes.
Thank you, sir.
Good morning, sir.
Good morning.
Good morning, sir.
Yeah, good morning.
Good morning, sir. My name is Saubhagya, sir. I'm studying
Okay.
first BSc Zoology, sir. My question is, what are various statistical tools used by International Rice Research Institute, sir?
Thank you, Saubhagya. So currently, actually, when we actually I joined in the 2017. So even when I worked with in during my PhD in National Rice Research Institute, Cuttack. So completely, I'm not aware of that any best way of analytical tools, because of we are mostly based on the Windows-based statistical tools. Like if you remember, there are tools like in a plant breeding tools, Star, Windows Start, or some kind of window-based application. Because of what is that, I'm going to collect the information from the Excel sheet. So once I open the applications, I upload the data. Then what I need to do, so just I'm selecting the variable like plant height, a grain yield. I want to do some correlations. Just to tick, tick, tick. Just all the do the click and you'll get the data in the graphical table informations. That I has to do in the during the page time. But when I work with this, one of my mentor is very strong in the quantitative genetics. He did his PhD and postdoc in Nebraska University. he did a lot of work on the data analytics. So then I started working on this 2022-21 onwards, R-based analytics, R program. So R program is one of the beautiful statistical program or statistical software, any scientific discipline, either your animal, plants, or research, pharmaceutical, or any software company, anything. Because our program having very flexibility to design or to do the analysis in a very systematic, because it is user friendly software. Currently we are also using the R program with using a different language packages. So modern packages, how to just use the package informations and do that analysis. So our program is one of the most currently we are using on the at ERI also. And the CGR is developing one kind of R based, like in Shiny based. If you remember 2023, one of the slide, R Shiny. So there's a Shiny based applications will be used in currently at ERI.
Morning.
Morning.
Thank you, sir. Good morning, sir. I am Ranimita.
Morning.
I'm from first BSc. My question is, sir, is there any student-oriented training programs at IRI, sir? Then how to apply?
Thanks, Pranvati, actually, for your interest on that. So actually, ERI is providing some kind of, I think, ERI sandwich fellowships for the particular for the UG and PG students. And I think there will be called NIPHM, National Institute of Plant Health Management. NIPH is particular for this institute will be collaborating with the ERI and Indian Institute, where the students will be migrating, or students will be going to the abroad, will explore some kind of a 3 months or 6 months program, and all of the funds, the expenses will be covered from the NIPH. And ERI also providing some kind of sandwich fellowship, but exactly from the, I'm not sure for the UG students, and PG students will be there. And yeah, these are some organizations will be there every year. Even last year, from the, I think from the Uttar Pradesh, Jharkhand, Assam, some UG students, almost of around 16 members or 12 members, they came to ERI for the 3 months for exploring the how ERI is working on the research areas. So there are some ways are available, but if anyone interested on that, if whatever the some have some links, I will share it with you if you connect with me if any in your free time, or maybe if you're interested on that. There are things are available for the exploring to the ERI fellowships.
Thank you, sir.
And also there is a Google, you can search in the Google ERI educations. So particularly there is a website ERI educations, that education team will helps to the students for exploring to the this kind of research programs in ERI education team. Yeah.
A very warm good morning, sir. No doubt
Yeah, good
your
morning.
lecture has been enlightened us, and we have made an insight in order to draw the better decisions. No doubt the quantitative genetics and the bioinformatics are the powerful tools. And my question is, you have told that you know, in order to overcome the challenges and to meet the global needs, we have to
Yeah.
prepare the new strains by using the modern technology in the plant breeding methods. So while we are producing the new varieties in the plants, does the strain has any impact on the human physiology? As in the present days, in order to get the milk throughout the year, the farmers are using the hormonal injections for the cows in order to get the milk throughout the year. So likewise, we are developing the most important varieties in order to overcome the drought conditions and all those things. So in doing that particular pattern, so we are altering the gene of the plant. So does that gene has any impact on the human gene as it is at the gene level?
Thanks. Can you hear me?
Pardon?
Hello. Can you hear me?
Does the altered plant gene has
Yeah,
impact on the
I
human gene?
I got your question because of my slides or my. I cannot say that. Can you hear me? My voice. Is it? Am I audible? Yes.
Yes, sir,
Yeah.
somewhat.
Yeah. Thank you. Actually, it's a very good, fantastic questions from your end, and you are very well capture this quantitative genetics in the plant breeding and animal breeding and correlating with this particular aspects of the milk productions related to the human health in the physiological point of view. Well appreciated for your question. Is very interesting. So I will give you some two important points. So thing is, when it coming to, because of this, the quantitative genetics came from the this modern breeding strategy, genomic selections is came from animal breeding program. So the animal breeding program is based on this, the milk productions or the meat productions. So these, as I mentioned, these are the major trait is governed by the quantitative traits. Quantitative traits, nothing but is not single gene, many genes. So the many genes is contributing to the particular one trait. So when you go for these genomic predictions, the quantitative genetics aspects, we are going to capture that particular for the additive genetic informations that can be contributing to the your particular trait. So we are capturing the genetic informations to improving that particular trait. So not impact on the any physiological impact. The key message here, this is not from the any, for example, any other breeding step. This is a totally conventional way of modern breeding strategies. How do we capture enough sufficient genetic information to build the better performance or cow or buffalo, which is giving that a higher yield, higher milk productions, or a high productivity of the meat. The ultimate our aim was to improve it. How do we improve it? Improve the, bring in the as much as possible for the very good genetic information. The genetic information is coming from the additive genetic informations to particular trait. That trait can improve, but does not impact on the other trait, other impact on the health, because of the milk is producing from this particular trait. That trait is contributed certain genes, but we need to bring all of the genes together to build it. So the direct answer is it's not impact on the any negative or impact on the physiological in the human health. This well established in the human genetics in previously. The similar models, the mixed models, how we are building the predictions made models are mixed models used in the animal breeding. Now we are bringing into the plant breeding community to bring that within the shortest way of to improving the higher yield. But that's all depending on there are several ways. As I mentioned, the breeder equations, the speed breeding facilities, the how we are using the data analytics. So these are all playing a key role on that. I hope I will answer further questions to you. Hello? Hello? Hello, can you hear me?
Good morning, sir.
Good
Thank
morning.
you, sir, for this session. I am
Thank
Jijoshna. I
you.
am studying second year BSc Zoology, sir.
Okay.
I would like to express my sincere gratitude to our eminent keynote speaker, Dr. Mahinder Anmula, sir, for delivering on insightful and expert talk on data-driven insights from quantitative genetics and bioinformatics to better decisions. Sir, session provided clear and in-depth understanding of how data-driven approaches, quantitative genetics and bioinformatics play a crucial role in modern biological research and informed decision making. The way complex concepts were explained with clarity and real world relevance greatly enriched the knowledge of faculty members, research scholars, and students. We are truly thankful for his valuable time, dedication, and willingness to share his expertise, which made this program academically meaningful and inspiring. Once again, we extend our heartfelt thanks to Dr. Mahendra Anmula sir for his enlightening and impactful contribution. Thank you, sir.
Thank you. Thank you so much.
Thank you so much, section, sir.
Thank you so much.
Morning, Mahinder. Thank you so much for the wonderful session. And you made a PPT which has been transferred from international to the local. Thank you so much. Everybody
Thank
understood it. Thank you.
Great, great. Thank you, Sivanjani, for giving that confirmation, because that's only one one reason I spent some time, because of I don't want to give this some kind of where I was expertise. I'm bringing this dump on that different ways, but for their understanding, just I'm giving this some insights. I hope you all like it, and let me know if you have any questions or any information further on it. So thanks for so much for this, principal ma'am, and for all the organizing committee members for giving this wonderful opportunity. Thank you all.
Okay, thank you. Next session will continue.
Now I request M. Mohan Tejaswini, second BSc Zoology, to deliver the introduction. Resource person, Nutalapati Srinivas Garu.
Good morning to everyone. It's my immense pleasure to introduce great teacher, high passionate in research and teaching, Dr. Nutalapati Srinivas Garu, lecturer in zoology of Government Degree College, Ramchandrapuram. He completed his PhD at Andhra University in zooplankton production and distribution in mangrove habitat of Godavari Estuary, Kakinada. He received Best Teacher Award for outstanding result in zoology subject. He attended 5 training programs, presented 12 seminar papers, and also acted as a judge for the 16th AP State Level Children Congress, Vijayawada 2008. He published international journals and author for the intermediate zoology government textbook for both first and second years in Telugu and English medium, published by Telugu Academy 2014, and also author for the intermediate zoology MSET question bank for first and second years, both English and Telugu media, published by Telugu Academy in 2015, and also author for APSCHE UZ bilingual textbooks in zoology. He prepared 58 LMS for APCCE and APSCHE. We are fortunate to have you, sir, and thank you for accepting our invitation for today's webinar. I extend a warm welcome to Dr. N. Srinivas Garu and invite him to take the session forward. Thanking you, sir.
Very good afternoon, Andy. Am I audible?
Sir.
Okay. Is the screen is visible to all of you and screen is sharing? Can I proceed further?
Yes, sir. It is visible, sir.
Okay. So I'll try to focus on certain aspects related to the genomic data and how the data has to be leveraged in decision making in healthcare aspects. So it may be not an academic aspect, but an aspect which is related to the present day scenario and present day scientific research. So I'll make this presentation a general presentation, and all the information is gathered by scientific literature. And the presentation is mostly for the students who are pursuing biotechnology, zoology, and aquaculture, their UG and PG. So my talk is basically focused on the future prospects for the students of the life sciences. At the outset, my sincere thanks to our beloved Commissioner Sir, Dr. Narayan Bharatagupta Garu, our beloved Joint Director Sir, Dr. C. Krishna Garu, and our Joint Director, Zone 1 and 2, Dr. P.V. Krishnaji Garu, and the host college principal, Madam Dr. P. Raghava Kumari Garu, and our college principals are beloved Dr. C.H. Ramakrishna Garu, and my predecessor speaker, scientist from International Rice Research Institute, Dr. Mahendra Garu, and the organizing this webinar organizing convener, Ms. C.H. Priyanka Garu, and organizing secretaries of this webinar, Deepika Rani Madam and Sivaranjini Madam, and also the members of the organizing committee, Divesri Garu, Rasma Garu, and Lilavati Garu. Thank you very much for inviting me as one of the resource persons for this international webinar. So, outline of my talk will be on these aspects. Firstly, in the first session or first few slides, I'll focus on the concepts of the genomics. Basically, we are traditional biologists. That means we will have our botany, zoology, chemistry, something like that. So this one, genomics, is a applied concept. So I'll try to give insights of the concepts of the genomics. Next, what are the different tools that are available for the genomics? Similarly, the applications of the genomics. Next, how genomics impact on the data-driven insights. Next, there are regulations by the governments as well as the research organizations. And what are the regulations and under which regulations these genomic concepts and these genomic activities or genomic applications are being governed. That will be given or that will be presented to you. Next, future research directions. So this is most important for all the students and as well as young faculty members who are working or who are having the zeal for applied sciences, from conceptual basic science to applied science or conventional science to applied science. That part will be there. Next, challenges and limitations. So my talk will be in a nutshell on these aspects. So firstly, I'll give a general introduction to genomics. Evolution of the genomic science. Genomic science has evolved from Mendelian genetics. We all know from our high school studies or intermediate studies, we know that Mendel is the father of genetics. So in 1970s, as I mentioned by the earlier speaker, Sir Mahidhar Anumala Garu, also he has mentioned the classical genetics. So 1970s onwards, DNA sequence to modern high throughput technologies, enabling the analysis of entire genome structural variations, evolutionary mechanisms like duplication and exon shuffling. So right from classical genetics to the applied genetics and the genomics, it has evolved. It has shifted from single gene analysis to understanding genome complexity, regulatory networks, and population level genetics. So initially, genetics focused on the study of a specific traits, as I mentioned by the earlier speaker also, Mahidhar Garu also. Traits, from traits to genomes. That means we are focusing or we are contemplating on the entire genome of an individual. So that is the beauty of the genomics. Next, genomic data. Genomic data matters because it unlocks personalized healthcare. Why we are very much fascinated or very much interested about genomics and genomic data? This genomic data unlocks the personalized healthcare. If you observe the healthcare sector in India or globally, that is a huge market having very rich economics and rich outcomes. So this healthcare sector allowing the doctors to diagnose, treat, and prevent the disease based on individual's unique genetic code. So if we have genomic data, the healthcare sector will be somewhat modified. That means the medicine or the application of the or administration of the certain types of medicines will be drastically changed. So that type of data is there in the genomics. By mapping DNA, researchers can develop targeted drug therapies, predict risk for complex conditions like cancer or diabetes, and improve patients' outcome. So the treatment will be somewhat better when we compare to the present scenario of healthcare sector. So that is the beauty of genomic data. Next, applications, challenges, and future trends. Genomic data is transferring healthcare from a reactive one-size-fits-all model. If you search the net, this is the buzzword one-size-fits-all. That means medicine is used to each and every person, irrespective of their body conditions or their nature of acceptance of that particular drug. That is known as one size fits all. Model to proactive personalized approach. That means a specific drug can be administered to an individual having specific disorder. That type of change will be there in the near future if we follow the genomics. Next, by leveraging technologies like next generation sequencing. This is the another word we mostly use while we are focusing on genomics, NGS, next generation sequencing, and AI. In the previous session also, some of the students posed the question, what is the role of the AI? So that plays also an important role. In healthcare systems can analyze a patient's genetic profile to improve diagnosis, treatment, and prevention. So these are the three important components, that is diagnosis, treatment, and prevention. For the purpose of diagnosis or diagnostic measures, the genomics can be useful. Similarly, for the treatment also, the data of the genomics can be used. Similarly, for the preventive aspects also, genomics plays an important role. Next, what is the difference between genetics and genomics? You can clearly visualize in this picture, genetic, a fundamental science of heredity, the building blocks, whereas the genomics, an encompassing modern field that uses genetics as a foundation but studies the entire system of genes as a whole. For example, if you consider the genetics, we will confine to a specific trait or specific character. Whereas in case of genomics, the whole nature or whole constituent genes of the individual can be studied. So that is the important characteristic feature of the genomics. So genetics means we confine to a special character, whereas or a specific character or a single character or a single trait. Whereas genomic, we have to focus on the entire genome of the individual, whether it is a plant or any animal. That means integrating all the characteristic features of that particular individual can be studied under genomics. Madam, the slides are moving, or madam, are anybody please respond once?
It's visible, sir.
Slides are the slides are moving, madam. History of genomics. Now the slide is history of the genomics.
Yes, sir, they are in.
So genomics, the basis for the genomics is the Human Genome Project, which started in 1990, and by 2003, it has revolutionized the genomics. The Human Genome Project was a 13-year-old international research effort, 13-year-long international research effort from 1990 to 2003 that successfully mapped and sequenced the entire human genome, comprising roughly 3 billion base pairs. So if you consider our human genome, it is consisting of 3 billion base pairs. It identified approximately 20,000 to 25,000 human genes, revolutionizing medicine, biotechnology, and forensic science by providing a gold standard reference sequence for genetic studies. So HGP, Human Genome Project, has given a breakthrough for this genomics. It has identified nearly 25,000 human genes having special characteristic features. And all these genes became the standard genes for any type of disease or character, something like that. So all the identified genes under Human Genome Project became the standard genes globally. Based on that reference gene, we can assess the nature of that particular gene in a patient or in an individual. So we can manage or manipulate that particular gene based on the observations of the genomic and correlating the standard gene with the observed gene. So that is the breakthrough in the genomics. Next, clinical genomics. Initially, human genomics was a great breakthrough. Later, the clinical genomics came into picture. Clinical genomics is rapidly transforming healthcare from reactive to proactive. As I have mentioned previously, one drug for all the individuals, or one medicine, or one antibiotic, something like that. So used by the doctors or the health practitioners to anywhere. But nowadays, it has changed. Leveraging the next generation sequencing to provide personalized evidence-based medicine. So these medicines, based on the individual's genomic characteristics and observations and reference with the standard, the medical practitioners as well as the doctors have differentiated the medicines into personalized medicines or evidence-based medicines. And driven AI integration, if you integrate these observations with AI, it enables precise cancer diagnosis, rare disease identifications, and prenatal screening. So these are the advanced characteristics based on the application of the genomics. We can able to diagnose cancer precisely. Previously, it is not possible. Now it is curable and also perfect diagnosis of cancer is done with the application of the genomics. And rare disease identification and prenatal screening. This shift is moving from specialized research to routine clinical practices essential for tailored therapy. So the therapeutics also has changed, and the individual is being given a specific drug to act at a specific site for treatment. So that is also changed due to the genomics. Next, coming to the types of genomic data. So what are the genomic data that is available readily for the scientific community? The first one is whole genome sequencing. Whole genome sequencing is a comprehensive laboratory process that determines the entire DNA sequence of an organism genome, including all chromosomes and mitochondria in one test. So WGS, whole genome sequencing, is a test where the entire genome of an individual can be sequenced. By capturing both coding and non-coding regions, whole genome sequencing identifies variations like single nucleotide polymorph, SNPS. So this is an important part in sequencing, insertions and deletions to diagnose rare diseases, characterize cancer, and track pathogens. So these are the focused areas while we are analyzing the whole genome. That means SNPS, insertions, and deletions. Based on these appearance in the genome or availability or randomly availability of these structures, specific diseases can be diagnosed easily. So this is the process of whole genome sequencing, genomic DNA fragmentation, next generation sequencing, and by data analysis. So this is the method. So whole genome sequencing. Next is the second one is the whole exome sequencing. Previously we have yes, madam. Previously, we have studied about whole genome sequencing. Now we focus on whole exome sequencing. Whole exome sequencing, that is WS, is a comprehensive genetic test that analyzes the protein coding regions, exomes of DNA to identify disease-causing mutations. So these are the specific areas in the genome which is having some mutations, and they result in specific diseases. These regions make up to only 1 to 2% of the entire genome. They contain roughly 80% of the known diseases related to variants. So this 1 or 2% of exome is responsible for causing 85% of different types of diseases in human beings. That means if we analyze this portion, that is 1 to 2% of the genome, that is exome, we can identify, predict, or diagnose different types of diseases, nearly 85% of the different types of diseases that are existing in the mankind. So WES is a cost-effective alternate to whole genome sequencing. When we compare to whole genome sequencing, we have to analyze the entire genome, whereas in WES, whole exome sequencing, only specific regions are sequenced. And our processing time is very less, and the result will be very clear. For that reason, it is used for diagnosing rare diseases, cancer, and developmental disorders. So by analyzing WES, we can predict certain rare diseases, cancer, and developmental disorders also. Next, targeted gene panel. Types of genomic data, we are continuing that. And this one is targeted gene panel. Targeted gene panels are specialized diagnostic tools using next generation sequencing to analyze a select set of genes or genomic regions known to be associated with specific disease, such as cancer and hereditary disorders. Again, this is also, if we know that specific portion of the genome is responsible for causing a disease, we particularly focus that particular region and sequence that region. That is known as targeted gene panel sequencing. That means we are confining, or we are narrowing down our processing time and sequencing time. Initially, we have sequenced our entire genome. Later, we have confined to exons only. We have sequenced. If you know the specific targeted gene panel is responsible for that particular disease, that gene panel is targeted and that is sequenced for further analysis and what we call as confirmation of the particular type of disease. So this is that means we are narrow downing our processing time as well as sequencing time. Next. So this is the protocol of the genomic data generation. That means we have to collect the sample either from blood or saliva of the individual. Next, we will extract the DNA by standard methods. Later, sequence with different, that means next generation sequencing, and the data output, that data is visualized or data is interpreted with the available data of the genomic database. So next generation sequencing, why this next generation sequencing, NGS, is more important? It is high throughput sequencing. That means the output is very high. often called as next generation sequencing or massively parallel sequencing, enables rapid, cost-effective, and simultaneous sequencing of millions of RNA, DNA, or RNA fragments. Within no time, the amplification of the specific targeted DNA or RNA is done through NGS. Next, another important concept or another important characteristic feature of the NGS is fast and cost-effective. So high throughput sequencing has made it possible to, for example, generate 1.8 terabases of data within 3 days using HiSeq X10 system. So the multiplication is very much faster by next generation sequence. And these are the different available sequencing instruments or sequencing processes in the market. The first one is Illumina sequencing. Next is nanopore based sequencing. Third one is RNA polymerase based sequence. By using these, the genome or the targeted gene or WS can be sequenced within no time. So this is the genomic analysis, genomic data analysis pipeline. How we have collected the genome or specific part of the genome, and we have analyzed it, and later we have to interpret that particular data. For that, this is the mechanism or this is the process. So raw data generation. Next, we have to align and variant calling. Annotations will be there, and later clinical interpretation will be there. All will be done by using the computer-aided programs. Next. So that is the, up to that point, I have given an introductory part of the genomics and how the genomics are playing a crucial role in the medical field nowadays. Now I am focusing on interconnecting the different fields of biology, applied biology, that is bioinformatics or biotechnology, with the genomics. So what is the role of bioinformatics in genomics? We all know that bioinformatics is computational analysis of genomic data. For the computational analysis of genomic data, we need bioinformatics. And bioinformatics uses algorithms and databases, essential for interpretation. For the interpretation of the genomic sequence you have done, with the genomic data that is already available, we have to have a bioinformatics or specialist in bioinformatics having thorough knowledge in bioinformatics. That is very much needed. Next, clinical genomics. We have focused on the healthcare sector and how the genomics play an important role in healthcare. There the picture of, there the role of clinical genomics comes into the picture. Applying genomics in patient care, diagnosis, prognosis, and treatment selection. All these are under clinical genomics. That means for the students, you have to remember that these are the future areas. Bioinformatics and clinical genomics are having very much potential job opportunities in the near future. Apart from your classical zoology or botany, basically we must have our zoology or botany. Along with those botany and zoology and chemistry, we must have a thorough knowledge on these applied components. Next, precision medicine. Tailoring treatment to genetic profile. Based on the profile of that particular patient, treatment can be designed, or treatment can be planned, dosage can be planned. Improves efficiency, efficacy, and safety of the patient. Reduces trial and error prescribing. For example, if you consider we have a fever, generally we go to the medical shop and buy paracetamol tablet and administer to the paracetamol. In precision medicine, it is not there. Based on the genetic sequencing or the output the doctor has received from your genomic information, he tailors the treatment. Or he generates a program so that, for example, if you have fever on X day, by Y day, this disease will be treated and cured, something like that. That much of precision will be there if we analyze the genome and analyze the existing genomic data and plan for the tailoring of treatment, something like that. Next, pharmacogenomics. This is also another important area where the administration of the drugs, study of gene-drug interaction. That means these drugs interact with these specific genes and predicting drug response. Similarly, based on the drug, the gene will respond to that particular specific drug, avoids adverse drug reactions. All these can be simulated with the available data. And based on the result of that particular simulation, the drug can be administered to the individual. That means there is no harm with regard to the, or no adverse effect with the administration of that particular drug. That can be visualized, that can be predicted before administering the drug to the individual. So patient genotype can be assessed and drug selection will be done, dosage optimization, reduced side effects. For example, if you consider there are certain drugs which cause side effects to the individuals nowadays. But if you apply or if you follow pharmacogenomics, that means the genomic data is there for specific drugs, for specific disease. By using that data, you can administer a specific drug to an individual, minimizing the side effects. That is the beauty of pharmacogenomics. Next, cancer genomics. This cancer genomic identified tumor-specific mutations, guides targeted therapy, and supports personalized oncology. Nowadays, the cancer research is very well advanced and mostly focused or mostly associated or mostly dependent on this genomic and genomic data. Why? Because cancer genomics identified tumor-specific mutations. That means the genes responsible for causing a tumor are being identified and that are there in the genomic data. By correlating the observed sequences with available data, we can predict that this particular gene is causing a cancer to the individual. Then we start the therapy for that particular gene only, which is causing or which is having mutation and responsible for causing cancer. That means the drug is specifically targeting that particular gene, which is under mutation, responsible for causing cancer. So that type of specific site-specific activity can be done with the available data in the genomics. So this is the process, that is tumor biopsy, mutation detection, target identification, personalized therapy. That particular target, for that particular target only, we can administer specific drugs based on, again, pharmacogenomics. So that is the beauty of the genomics. Next, inherited genetic disorders. For example, cystic fibrosis and Down syndromes. These diseases can be detected at the earliest stages, prenatal stages, and intervention can be made by approaching different techniques that are available in the genomics. So these disorders, which are hereditary, That means these disorders can be transmitted from parents to the offspring, can be prevented at the early intervention by the application of the genomics. Next, preventive care. Genomics in preventive care. Risk predictions for disease. Lifestyle and monitoring of recommendations. Early screening strategies. All these are for preventive care. Prevention or early detection of different types of diseases based on the genomic sequencing or genomic makeup of the individual. So we can address those things by studying the genomic makeup of the individual. Next, population genomics. This is the study of genetic variations in population, supports public health planning, and improves diversity in research. So this is the one of the area which is having potential for research, diversified research. We are focusing on the population genomics, not an individual genome, population genome. Okay. We can study the variations in the populations and support the public health planning with the observations and improves the diversity in research. It moves beyond single gene studies to examine genome of wide patterns. Individuals are different and genomic patterns are quite different, aiding in studies of human ancestry, disease susceptibility, and species adaptation. How the disease is still sustaining, and how, even though diseases are sustaining, how the species are adapting to that particular susceptibility of the disease, that can be studied with the population genomics. Similarly, genomics in infectious diseases, pathogen sequencing, tracking outbreaks, antimicrobial resistance detection. So these are some of the areas in genomics in infectious diseases, application of genomics in infectious diseases. Pathogen genome can be sequenced, and based on the pathogen sequencing, you can track the outbreaks. For example, in a specific area, say for example, X, in a X area, the outbreak is there. Then we can assess based on the environmental parameters, this particular outbreak will be expected at Y area or Z area, something like that can be assessed. That is known as tracking of outbreaks. That can be done with the genomics in infectious diseases. The nature and the genomic tendency of that particular pathogen can be tracked, and we can take preventive measures to prevent the infectious diseases based on the genomics in infectious diseases. Next, there are some ethical issues also there with regards to genomics. Privacy of the genetic data. We have huge data of the genomic data of the individuals or something like that. So that should be important to the individual. So that should be considered while we are analyzing or dealing with the genomic data of the others. Next, informed consent. We have to take consent from that particular individual when we are mapping or when we are studying that particular genome of that particular individual. Next, genetic discrimination. If the traits, if you consider the traits and the qualities, something like that, discrimination will definitely creep into picture. Why? Because every individual or every human being or any animal, it totally relies on the quality. We need quality. We need good quality or better quality or the best quality. So in each genome or each individual, we cannot expect the best and the better qualities. So the qualities with the best qualities, genomes with the best qualities will be targeted. So that is the main problem or main ethical issue, misusing something like that. We all know that what are the ethical issues nowadays in scientific research and paper publications. All those creep in when we consider the ethical issues related to the genomics. Privacy and genetic data, informed consent, and genetic discrimination will be there when we talk about the ethical issues in genomics. Next, legal and social implications. There are some legal and social implications. Number one is data ownership. The data is very huge, and for the purpose of ownership of that particular data, legal and social problems will be there. Similarly, insurance, employment concerns, and regulatory frameworks are some of the concern with the implications of the genomic data. Next, genomic data privacy. Data should be secure, controlled access, and patient confidentiality. Why? Because the key protection measures include encryption, de-identification, access controls, and privacy by design, often supported by the regulations like GINA and GDPR. So these are the two regulatory bodies available in the world nowadays. Number one is GINA, Genetic Information Non-Discrimination Act. Number two, General Data Protection and Regulation. Both govern the sensitive data but have different scopes. GINA is a US law preventing genetic discrimination in health insurance and employment. Whereas GDPR is EU regulation protecting personal data privacy. GDPR offers stronger protection for genetic data as a special category. So these are the privacy regulatory authorities globally. Number one is US based, another one is EU based, European Union based one. Next, some of the challenges in genomic medicine. High data complexity. When we want to compare the available genomic data with the genome library, it is very complex. And interpretation difficulties. Since we are relying or depending on the computational aids and computational tools, there is a difficulty. Some difficulties will be there. And limited trained professionals. So this is the another important area where the youngsters have to take a call. Limited trained professionals. If you are well versed or well trained in genomic studies, bioinformatics, something like that, you will be paid very much in the near future. So the key hurdles include interpreting variants of unknown significance. Our genome consists of variants also. So while analyzing these variants, it is very difficult for the interpretation because we don't have a reference for that particular unknown variant, unknown significant variant. Similarly, keeping up electronic health records, EHRs. This is also another area where in near future, all your individual health records are kept as kept in electronic format, EHRs, electronic health record. Based on that, only in future treatment or diagnosis and everything will be there. So some of the hospitals or corporate hospitals may be there having or maintaining these EHRs of every individual. Nowadays, our government has also planned, and a few days back, our chief minister also analyzed that the healthcare of the individuals will be taken care of by the government by using AI. So that is the mechanism they are employing, EHRs, keeping up electronic health records of the individuals of that particular area. Next, limitations of the genomic data. Variants of uncertain significance, incomplete knowledge, and population bias. So these are the certain limitations where we cannot predict or we cannot assess or we cannot conclude a confirmation whether it belongs to X or whether it belongs to Y, something like that. So these are the variants of uncertain significance, incomplete knowledge, and population bias. Next, genomics in healthcare systems. In near future, we can visualize or we can experience this integration into clinical workflows, electronic health records, and decision support tools. Once you give the genomic data of individual, all these will be generated spontaneously at the healthcare systems or health supervisors or by the health supervisor or health assistants or doctors, something like that. Next, role of AI in genomics. So this is our point of interest: pattern recognition, variant interpretation, clinical decision support. So artificial intelligence also plays an important role in genomics. AI in genomics accelerate the data analysis. So we have data, we have referral data, we have the patient data. These two things have to be analyzed, and the similarities, variations have to be projected immediately by the system. At that point of time, the role of AI is very much important and it plays a crucial role. So that is explained here. AI in genomics accelerated data analysis, interpreting vast data sets from NGS, next generation sequencing to identify disease-causing variants and enable personalized medicine. It enhances drug discovery, optimizes gene editing, CRISPR-Cas, you might have heard about that CRISPR-Cas gene editing, predict disease risk via polygenic risk scores, and improves diagnostic accuracy for rare diseases. If you think of certain diseases which are very rare in mankind, those can be treated with the genomic data. Next, the role of machine learning also very important in genomics. Machine learning plays an important role in disease risk prediction, probability prediction. Next, similarly, drug discovery. We already have existing drugs. However, the combination of the drugs or administration of the drug or assigning a specific drug to that particular genetic modification or gene mutation can be done by ML or genomic classification. All these are done through machine learning applications. Next, these are the genomic databases now available globally. First one is GenBank, US based one. Next, CleanVar, that is also US based one. Next, 100 Genome Project, 1000 Genome Project, sorry. The 1000 Genome Project was a landmark international research effort that created the most detailed catalog of human genetic variations by sequencing genomes of over 2,500 individuals. Genomes were identified, and all the genome is mapped and kept in that particular genomic bank, genomic database for reference. Next, in research, how the clinical genomics is useful. Discovery focus. That means already existing drugs are there, existing diseases are there, or existing therapeutics are there, everything is there. But we can focus on the variations, small variations that are there in the genome. And based on those variations, we can perform different types of analysis, which makes a new research or new intervention. So that's why clinical genomics is very much useful for research. Next, this clinical genomics is patient-centered. Nowadays, most of the hospitals or most of the doctors prefer specific drug because that drug is manufactured by some pharmaceutical company. That type of tendency will not be there in near future. Next, different standards and regulations that will be quite different based on the clinical genomics. Next, in education and training also, genomics are also useful. Need for genomic literacy. We all know that the genome of the individual and how the genome is identified and how the genome is sequenced and what role is playing in genome in our individuals, our trait, or our characters, or something like we should know. Next, interdisciplinary training. We must have thorough training in interdisciplinary aspects, something like I have mentioned bioinformatics, genomics. Next, we have also computational analysis or software, something. We have to learn and we have to know, or at least we have to know the basics of those components for understanding the genomics in near future. Faculty and students should be engaged in training on these genomics and genomic concepts. Next, role of health professionals, clinicians, genetic counselors, bioinformaticians play an important role in the genomics and genomic studies in near future. Next, genomics plays an important role in cancer treatment, as I have mentioned earlier in earlier slides also, tumor sequencing, targeted therapy selection, and improved outcomes. Next, in maternal health, prenatal screening, carrier testing, and risk assessment can be done by genomic studies in the maternal health. So this is the future of genomic medicine, protein genome sequencing, integration with AI, personalized prevention. A day will come in near future, a drug will be specifically given to a targeted gene. So that is the near future with the genomic and genomic medicine. Global genomic initiatives, international collaborations we need data sharing, and we need equitable access to the genomic data that is already there existing in the globally at different genomic banks or genomic databases. Next, decreasing sequencing costs. This is already done, but still the price has to be again decreased for to reach the common people. And access disparities, policy considerations. So these are the some of the challenges while we rely or while we depend on the genomics. Next, these are the regulatory bodies of the genomics. First one is FDA, US Food and Drug Administration, EMA, European Medicines Agency. Coming to India, national genomic policies. Different countries have formulated their genomic policies. For example, our India, the Central Drugs Standard Control Organization, CDSO, has notified new drugs and clinical trials rules 2019, defining gene therapy products as new drugs and framing national guidelines for their development. So India has also formulated certain policy with regard to the use of genomics and genomic databases for the genomic therapy or gene therapy. So genomics also plays a role in the decision support system. We have our topic is also decision support system. So genomic alerts, treatment recommendations, and real-time support. If we know the nature of that particular gene, and we can assess or we can predict that the action of that particular gene, based on that, we can program genomic alerts as well as treatment recommendations and real-time support to the patient. Next, translational genomics. So this is the one of the important area where the research is to be reached to the patient, bench to bedside. That means from lab to land, something like that. We have a notion, lab to land, like that, bench to bedside, bridging research and care, implementation science. That means the genomics is mostly for the healthcare sector. I have mentioned in my talk earlier. If you consider the healthcare sector is more important or medicine is more important, our research should reach the patient. That means bench to bedside. Similarly, bridging research and care. if we carry out research at the laboratory level, it should be translated to the care of the patients. Patients having diseases, something like uncurable diseases. Nowadays, there are no uncurable diseases because of the advent of the genomics. Something like cancer, something like. So that can be useful for the care of the patient. Similarly, implementation of the science. Whatever the technology or scientific aspects we have in our laboratories, that should be implemented at the ground level for the common man. Next, this is another concept, that is multi-omics integration. In the genomics, the advanced concept is multi-omics. Genomics, proteomics, metabolomics. We have in the first or initial, at the beginning of my talk, I have mentioned that I am narrowing down from broader area to narrow area, something like that. So from genomics to proteomics to metabolomics, some similarly holistic understanding. Better predictions will be there if we follow the multi-omics integration of that particular patient or that particular individual. Next, genomics and personalized prevention, risk stratification, lifestyle interventions, and early monitoring. If you consider the genomics of the individual at the personal level, that at the individual level, we can stratify that particular risk of that individual by any type of disease or any type of pathogen, something like that. Similarly, lifestyle interventions. For example, if I am having obesity, based on that my genomic analysis or genomic sequencing or my genomic data, we can predict that, our doctor can, or a medical practitioner can predict that. So these are the consequences in near future to you, sir, based on our genomic analysis. So we have to take something like, you have to go for walking, or you have to go for exercise, physical exercise, you change your food habits, you change your lifestyle, something like that. So all those predictions will be there based on the genomics and personalized prevention. Similarly, early monitoring of heart attacks and cancer and some of the chronic diseases can be detected with the genomic studies. So this is the need of the hour, public awareness and education. Each and every student, each and every individual must have a genetic literacy. What is a gene? How the genes are responsible, and how the genes are responsible for individual traits, and what are the different characters, how the genes are interpreting, or how the genes are having an interplay in our daily, day in and day out metabolic activities. All those have to be given and clear understanding to the individuals. At least basic concepts of the role of the genes have to be given to the individuals. Next, community engagement. While we are giving an awareness, community should be engaged. Next, trust in genomics. Everybody has to give importance, or the role of the genomic has to be given prioritized or given much preference, because they are highly useful and they are specific to the individual. So that is the need of the hour, public awareness and education. Next, this is also another area of the interest, research acceleration, standardization, and ethical data usage are to be given importance in data sharing and collaboration within the countries or outside the countries or within the institutes or among the institutes or among the researchers. That is also the need of the hour. Next, sustainability in genomic medicine. If we want to achieve a sustainable genomic medicine, we need infrastructure. Nowadays, the state of art technology and technological aspects and laboratories are very much less in our country. That should be focused. Next, long-term data storage. We are having billions and billions of databases, and the storage is a problem. We have to focus on the long-term data storage facilities for data interpretation, data retrieving. So that should be focused. Next, workforce development. So this is for your young students. If you have a knowledge on the analysis of genomic analysis or data interpretation, or retrieving the data, or matching the data, or mapping the data based with the available computer software, you will be paid very much. And this is also a focused area or emerging area for the new generation, workforce development. For example, sample collection, sample analysis, again, sequencing, sequence interpretation, mapping this existing sequence with the available data, and retrieving the data from the database. All these things has to be done by individual, by human being, by operating a specific machine, a computer, something like the software. But you are needed. Human being, intervention of the human being is needed. That is the need of the workforce. That is the target, or that is the challenge for the youngsters to focus on the importance of the study of the biological sciences, either botany, zoology, along with the applied sciences. Next, from my talk, these are the important key takeaways. I have mentioned the genomics and how it transforms the healthcare. And next, supports the informed decision making. Genomics is supporting the decision making in healthcare system. And this is one of the future ready discipline. This is one of the area where having lot of opportunities to the individuals, young ones, or graduates or science graduates. This is the area where very much opportunities are there, and try to learn or try to acquire the necessary skills to future ready graduates having the skills in genomics and analyzing the data of the bioinformatics or genomics, something like that. To conclude, the genomic data empowers clinicians. My focus is on healthcare. So the data of the genomics that is available, it gives an empower to the clinicians, either doctors or healthcare sector, healthcare supervisors, everyone, and improves patient's outcome. So the patient will be benefited and requires ethical and responsible use. However, the use of the genomic data should be under responsible and ethical use conditions only. If it is not used responsibly and it is not used ethically, it may lead to another type of devastation in the society or the community or the population. That is also our responsibility as citizens, as individuals, as educated students. We have the responsibility to use the existing data, genomic data, ethically and responsibly for the welfare of the patients at the risk of any type of disease. Next, these are the references I have used for this presentation. Now the session is open for questions. Over to the organizers.
Thank you, sir. Now the section is open for questioner.
Good afternoon, sir. My name is Tejashwini, sir. I am studying second BSc Zoology. Sir, today my question is how to increase diagnostic accuracy by using AI, sir.
Pardon, madam. How to increase
accuracy by using AI, sir?
There are different types of softwares, madam. Nowadays, we are in our country. We are using only prototype of softwares, R software, something like in the previous talk also SAR has mentioned. So there are different types of software. These softwares are using the AI applications of AI, and they integrate this software with AI. So algorithm, something like that. So they use the AI for giving interpretations. So only software based something. For example, if you consider your meta AI, if you give a question, it will generate some answer, something like that. Similarly, the AI is based on the data that is available in the system. If we have the genomic data, AI, you will give a prompt. Based on that prompt, the result will be there. So for AI, interpretation of AI, we need data. Okay, madam?
Okay, sir. Thank you.
సార్ పర్సనలైజ్డ్ మెడిసిన్ అన్నారు కదా, అది ఇంకొంచెం
Yes, sir.
చెప్పరా?
ఓకే సార్. సార్ చక్రపాణి సార్, గుడ్ ఆఫ్టర్నూన్ సార్. సార్ ఫర్ ఎగ్జాంపుల్ ఇప్పుడు మనం తీసుకోండి సార్, పర్సనలైజ్డ్ మెడిసిన్ అంటే ఏంటంటే, ఫర్ ఎగ్జాంపుల్ ఇప్పుడు నాకు ఉంది, నిన్న చెప్పాను కూడా, ఎగ్జాంపుల్ కూడా చెప్పాను. ఫర్ ఎగ్జాంపుల్ ఐ హ్యావ్ ఫీవర్, నాకు జ్వరం వచ్చింది. జ్వరం వచ్చినప్పుడు నేనేం చేస్తాను, డోలో 650 మన భాషలో చెప్తే పారాసెటమాల్ వేసేసుకుంటాను. వేరే పర్సనలైజ్డ్ మెడిసిన్ తీసుకున్నప్పుడు పారాసెటమాల్ అనేది 500 mgనా 250 mgనా ఒక రోజు వేసుకోవాలా, 2 రోజులు వేసుకోవాలా, 1 mgనా 2 mgనా స్పెసిఫిక్ గా మీకున్న, అంటే ఇప్పుడు మనకి జ్వరం అనే దానికి క్వాంటిఫికేషన్ అంటే మనం థర్మామీటర్ లో చూస్తాం. వేరాజ్ మనము ఈ జినోమిక్స్ ను ఉపయోగించి జ్వరాన్ని కనుక ఐడెంటిఫై చేస్తే, ఈ జ్వరం అనేది అది ప్యాథోజన్ వల్ల వచ్చిందా, నీకు స్ట్రెస్ వల్ల వచ్చిందా, నువ్వు ఫిజికల్ ఎక్సర్సైజ్ వచ్చిందా, మెంటల్ స్ట్రెస్ వల్ల వచ్చిందా, నువ్వు డైట్ లో తేడా వల్ల వచ్చిందా, అది కూడా చెప్తుంది. చెప్పినప్పుడు మనం ఏం చేస్తాం, అప్పుడు స్పెసిఫిక్ గా దీనికి ఎంత క్వాంటిటీ మెడిసిన్ ఈ రకంగా తీసుకో, ఇన్ని రోజులు తీసుకో అని చెప్తాను. సో అకార్డింగ్ టు, అంటే ఈ నా రిఫరెన్స్ ప్రకారం నేను తీసుకున్న దాని ప్రకారం అది అలా వస్తుంది అనుకుంటున్నాను. పర్సనలైజ్డ్ మెడిసిన్ అంటే అందరికీ రొటీన్ గా ఒకటే కాకుండా స్పెసిఫిక్ గా క్వాంటిటీ, సిమిలర్లీ టార్గెట్, అది పర్సనలైజ్డ్ మెడిసిన్స్.
Good afternoon, sir. My name
Yes,
is Varnakamla. I am
yes,
third
madam.
BSc. I am studying third BSc Zoology. Delivery is of
మేడం ఆడియో క్లియర్ గా రావట్లేదు, ప్లీజ్ ఒకసారి ప్లీజ్ చెక్ యువర్ ఆడియో మేడం, ఇట్ ఈస్ బ్రేకింగ్ అండ్ ఇట్ ఈస్ నాట్ క్లియర్ ఆల్సో.
delivery of software, sir.
మేడం ఐ డిడెంట్ గెట్ యూ మేడం, ఆడియో అనేది రాలేదు మేడం.
Elinvar is free or so paid software, sir.
మేడం ఒకసారి ప్లీజ్ ఒకసారి చెప్పండి అమ్మా, అంటే మీరు ఆడియో నాకు రావట్లేదు, మీరు ఏదో ఎలిన్ అన్నారు, అది ఒకటే వినపడింది తప్పితే మిగతాది రాలా.
Yes, sir.
ప్లీజ్ ఒకసారి చూడండి అమ్మా, ఆడియో ఇస్ నాట్ క్లియర్, నేను ఐ డిడెంట్ గెట్ యూ, ఒకసారి ప్లీజ్
Good
చెక్.
afternoon.
Yes madam, it is audible, very much audible, carry on.
Good afternoon, sir. My name is Lakshmi Saraswati. I am
Sir,
studying third BSc Zoology honors.
ఇంకొక విషయం,
2026 Indian healthcare sector union 2026-2027 still emphasizes strengthening rural and infrastructure by granting 1 lakh crore rupees. Then when we will get allocations for genomic medicine?
It is policy matter madam, it is not in our hands. As a people, as a public, that means we that is our responsibility. We have to give representation to the individuals or representation to the government, so that the genomic medicine will be given much importance in their planning. అంతే కదమ్మా, మనం చేతుల్లో లేదు కదా, ప్లానింగ్ చేసేవాళ్ళు వాళ్ళు గవర్నమెంట్ కాబట్టి మనం ఏం చేయాలి, మన చేతుల ద్వారా పబ్లిక్ రిప్రజెంటేషన్ కి ఇవ్వాలి. మాకు జీనోమిక్ మెడిసిన్ వాడండి, మామూలు మెడిసిన్ వద్దు, మామూలు హాస్పిటల్స్ వద్దు అనేది మన రిప్రజెంటేషన్ ఇవ్వాలి. దట్ కెన్ వీ కెన్, సో బేస్ యూజ్ దిస్ ప్లాట్ఫామ్, you make a resolution. We have conducted a webinar, international webinar on so day, and we have studied on the genomics and the importance of genomics. All the participants in this webinar has resolved unanimously that the genomic medicine should be given much importance to the rural areas. And a statement తీసుకుని you sign, all the faculty members and all the participants can sign and give it to the press. That becomes an event, and that becomes a point for the government. అలా చేయండి మీరు. సార్ చక్రపాణి సార్ ఏదో అడుగుతున్నారు చెప్పండి సార్.
Thank you,
మేడం
sir.
ఆర్ యు అగ్రీ విత్ మీ మేడం? మీరు అగ్రీ అవుతారని చెప్పింది పాయింట్ తేజస్వి గారు. ఓకే చక్రపాణి సార్ చెప్పండి సార్.
Thank you, sir.
ఓకే సార్ ఆడియో ఒకసారి చెక్, ఆడియో బ్రేక్ అవుతాను సార్.
ఇప్పుడు 1 minute సార్.
ఎస్ సార్ క్యారీ ఆన్ సార్.
ఏం లేదు సార్, ఇప్పుడు నోట్స్ రాయాలని అంటే చాట్ జీపీటీ కొట్టేస్తున్నాం.
అవును
అలాగే పిల్లలు కూడాను చాట్ జీపీటీ తోటో జెమిని దాంతోటో అసైన్మెంట్లు అవి టప్ప 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 రాసేస్తున్నారు.
ఎగ్జాక్ట్లీ.
అలాగే డాక్టర్గా కూడా నేను ఏఐయో లేకపోతే ఈ రెడీమేడ్ గా వస్తున్నాయి కదా
అవును సార్.
అవి వాడుకుని నేను ప్రిస్క్రిప్షన్ కి వెళ్ళిపోతే వైద్యం ఏమవుతుంది? జస్ట్ సీకింగ్ యువర్ ఒపీనియన్ సార్.
ఎస్ సార్ ఎగ్జాక్ట్లీ. భవిష్యత్తులో ఇప్పుడు రోబోటిక్స్, ఇప్పుడు సర్జరీ మీకు తెలిసిందే. నవ్ డేస్ ది సర్జరీ ఇస్ డన్ బై రోబోటిక్స్ ఓన్లీ, నాట్ ఇండివిజువల్. సిమిలర్లీ మోస్ట్ ఆఫ్ ది డయాగ్నోస్టిక్స్ ఇప్పుడు డాక్టర్లు ఎవరు చేసిన డయాగ్నోస్టిక్స్ అని బేస్డ్ ఆన్ ది టెస్ట్ని బట్టి వాళ్ళు చెప్తున్నారు. అంతేగాని వీళ్ళు ఏమి చేయట్లా. ఇక్కడ కావాల్సింది ఏంటంటే స్కిల్ అండ్ ఎక్స్పర్టైజ్ కావాలి. అది ఉన్నా కానీ స్కిల్ అండ్ ఎక్స్పర్టైజ్ ఉన్నప్పుడు మాత్రమే దానిని పకడ్బందీ. ఇప్పుడు ఇంతమంది ఇన్నిసార్లు వైద్యం చేయించుకుంటున్నా, ఇన్నిసార్లు మళ్ళీ వాళ్ళు దాన్ని ఏమంటారు సాక్రిఫైస్ చేస్తున్నారు, అది అయిపోతుంది ఇది అయిపోతుంది చనిపోతున్న కారణం ఏంటి? అక్కడ స్కిల్ ఎక్స్పర్టైజ్ లేని కారణంగానే ఎన్ని ఉన్నా కానీ దట్ ఈస్ ది ల్యాక్యునా. అది కావాలి మనం. ఇవన్నీ ఉండనే ఉన్నాయి, ఏ ఉండండి, ఇంకోటి ఏ కూడా ఉంది. ప్రాంప్ట్ అనేది సరిగ్గా ఇస్తే కదా మన అవుట్పుట్ సరిగ్గా వచ్చేది. సిమిలర్లీ యువర్ ఎక్స్పర్టైజ్ అండ్ యువర్ ఎక్స్పీరియన్స్ హోల్డ్స్ గుడ్ ఈవెన్ దో దెర్ ఇస్ ది ఇంటర్వెన్షన్ ఆఫ్ ది ఏఐ అండ్ ఎవ్రీథింగ్. అది ఇంపార్టెంట్ సార్. అది కొంతమందికే ఉంటుంది, ఆ కొంతమంది సక్సీడ్ అవుతారు అంతే సార్. హోప్ యూ అగ్రీ విత్ మీ సార్.
అంతే సార్. అంటే జుడిషియస్ యూజ్ ఆఫ్ దీస్ రిసోర్సెస్ కావాలేమో.
అది కావాలి. ప్లస్ సిమిలర్లీ దీంట్లో కావాలి కదా, యూ హ్యావ్ ఏ డెప్త్ నాలెడ్జ్ ఆన్ దిస్. అది లేకుండా ఎవరు పడతాడు? ఏ అందరూ వాడుతున్నారు. దాన్ని ఏమంటారు, మన మెటా దాంట్లో ఉంది మొబైల్లో, వాట్సాప్ లో, అది వాడుతున్నాం మనం. చాట్ జీపీటీ అందరూ వాడుతున్నాం. బట్ ఎంతమంది దాన్ని పర్పస్ఫుల్ గా బెటర్ బెనిఫిట్ ఆఫ్ ది సొసైటీ కోసం వాడుతున్నారు అనేది దట్ ఇస్ ది క్వశ్చన్. అట్లా
సార్. సార్ థ్యాంక్ యూ సార్.
ఓకే సార్.
Sir, a very warm good afternoon. And my question is, in the present scenario, all the parents were aware of stem cells and they are storing the neonatal stem cells in the stem cell banks. So, based on the pedigree analysis, so why can't we prevent the immunological disorders before the person is affected by drawing the genomics at the earlier stage?
It is there, madam. It is there. In my talk also I have given prenatal analysis. I have also mentioned that one. It is there, madam. It is there.
Is it there, sir?
It is there. It is there. It is there. It is possible and it is there also. Prenatal genomics I have mentioned in my something like 20th slide or 21st slide. I have mentioned that also.
Okay. So based on that particular genomic analysis, so that we can prevent the disease at the earlier stage.
Exactly, madam. Exactly. It is there. It is existing also. Nowadays, some of the celebrities and some of the people which are having high potential of affordability, they are using that technology.
Okay. Thank you, sir.
You might have heard about designed babies, something like that. డిజైన్డ్ బేబీస్ అనే మాట అన్నారు కదా మనం. అంటే కొన్ని మూవీస్ లో కూడా వచ్చింది అది స్టిల్ ఇట్ ఈస్ కంటిన్యూయింగ్. అంటే మనకి ఎలా కావాలి, బేబీ ఎలా కావాలి, కలర్ ఎలా ఉండాలి కూడా అదంతా కూడా దట్ ఈస్ నథింగ్ బట్ బేస్డ్ ఆన్ ది జీనోమిక్ ప్రిడిక్షన్, జీనోమిక్ మ్యాపింగ్. దాన్ని బట్టి వీళ్ళు మ్యానిపులేట్ చేస్తున్నారు.
Any questions from participants?
Thank you. Thank you for the session, sir. I would like to express my sincere gratitude to our eminent resource person, Dr. N. Srinivas Garu, for sharing his expertise talk with us today. Such insightful and comprehensive talk on the history and types of genomics, its wide-ranging applications, challenges, future trends, and the amazing role of artificial intelligence in genomics was truly enlightening and thought provoking. Thank you so much, sir.
Thank you, madam. Thank you.
Thank you for your sir. Now the webinar is concluded with vote of thanks by the Department of Zoology in charge, Ms. C.H. Priyanka, madam.
Good afternoon to everyone. First of all, I would like to thank Dr. N.B. Gupta sir, Commissioner of Collegiate Education, Government of Andhra Pradesh, Dr. C. Krishna sir, Joint Director of Government of Andhra Pradesh, Dr. T.V. Krishnaji sir, Regional Joint Director, Zone 1 and 2, for giving us this opportunity to organize international webinar. I extended my thanks to our beloved principal, Dr. P. Raghav Kumari Madam, for her encouragement and continuous support to organize this webinar. I extended my thanks to today's keynote speaker, Dr. A. Mahinder sir, for his depth of knowledge, greatly enriched our understanding and added immense value to this program. I would like to sincere gratitude to Dr. N. Srinivas sir for his continuous support and expertise talk. Thank you, sir. You inspired both the faculty members and students. I extended my thanks to organizing secretaries, Dr. Deepika Rani Madam, Dr. AKP Shivaranjani Madam, for their continuous support and cooperation teamwork. I extended my thanks to G. Divyasri Madam for her commitment. I extend my thanks to today's webinar committee members, Srimati M. Kasma Madam and Srimati Leelavati Madam, for their support. Thank you so much.
Okay, madam. Thank you, madam. I am signing off, madam. Thank you, madam. Thank you so much.
Thank you, sir.